Hello, I'm grateful for the opportunity to introduce you to Envirotherapeutics. I'm Neil Bhowmick, the CEO of Envirotherapeutics and professor of medicine and director of the cancer biology program at Cedar-Sinai Cancer Center. John Yu is my partner, and he's the vice chair of neurosurgery at Cedar-Sinai with experience taking cancer therapeutics to the FDA through public companies. This company currently has a couple interventions at different stages of development for cancer. Carotuximab ENV105 was our first, a neutralizing antibody that inhibits bone morphogenic protein, or BMP, through the CD105 receptor on fibroblasts. We're opening a phase 2 trial for prostate cancer and a phase 1 for non-small cell lung cancer this year. But what I want to discuss today is ENV205, a new therapeutic that addresses the unmet need of chemotherapy resistance and cachexia. We're looking to translate compelling preclinical work with your support. Many solid tumor patients are treated with chemotherapy. Unfortunately, all patients eventually have to stop taking the chemotherapy because they either develop resistance, have toxicity side effects, or have this muscle wasting disease called cachexia, which leads to the high mortality from cancer. Cachexia affects 20% of cancer patients, but for lung and pancreatic cancer, it's reported as high as 80% of these patients. Cachexia has long been recognized, but there's very little known of the etiology of this disease. It is defined by a loss of 10% body weight in 1 year or 5% in 6 months. Importantly, cachexia is not just a comorbidity of cancer, it's also found in renal cancer, in renal failure patients, lung disease such as COPD, and AIDS patients, contributing to well over $700 million of cost based on ICD-9 codes in the US alone. Both the US and Europe have designated cachexia as an orphan disease. Current treatments include appetite stimulants like marijuana and its derivatives, as well as steroids, and these mainly address the symptoms rather than the root cause. We published last year that mitochondrial DNA is elevated in prostate cancer patients on docetaxel chemotherapy, and it contributes to the chemotherapy resistance, where oxidized mitochondrial DNA coming from the cancer cells initiates an inflammatory cascade long identified in markers of cachexia as well as mediators of chemotherapy resistance. We have since shown a similar mitochondrial DNA secretion induced by other chemotherapeutic agents on relevant cancer types, including lung and pancreatic cancer. We developed ENV205 as an engineered antibody that binds mitochondrial DNA with high affinity and targets it to the liver by the hepatic endothelial Fc receptor for excretion, as depicted by the fluorescently labeled ENV205 at the bottom right. We hypothesized that the very same oxidized mitochondrial DNA that contributes to chemotherapy resistance also eventually leads to the muscle wasting as it makes its way to the skeletal muscle in cancer patients. To be able to perform the structural and animal studies, we needed to scale up production of ENV205 very early on in this research. We achieved this through the expression in Bacillus subtilis. We are seeking funds to develop the best formulation for ENV205. The obvious setting would be the IV infusion of the ENV205, likely in a similar manner to what we do for ENV105, which would have similar kinetics. The other would be to immobilize ENV205 on a solid support where blood would be passed through to deplete the patient of mitochondrial DNA. While this will require some strategic partnerships for the equipment, it would be the easiest in terms of regulatory hurdles, as no drugs would enter the body. And for the patient, it would be like getting a kidney dialysis. The third platform would be to introduce ENV205 producing bacillus bacteria itself in capsule form for a patient to swallow. As ENV205 is being generated from food grade bacillus right now, this would be the most direct delivery method. In summary, our company develops therapeutics that target the tumor microenvironment. The maturation of ENV205 in our pipeline is more than just another drug supporting chemotherapy sensitivity. It is a strong candidate for major unmet need in cancer and non-cancer patients. Because cachexia has an orphan designation, moving this forward will provide market exclusivity. Thank you.